time scales are completely different from the previous. Really. Okay, I'm presenting some work that I've been doing with my collaborator Xavier Didelot at Imperial College London, and this is the acknowledgement and founding, because there will be no time after. How do you move this? Well, okay. And there is a vast amount of literature on and methods that help us to infer evolutionary parameters such as the effective population size, or the mutation rate, or the expansion rate of expanding population. What we are doing now is dealing not with entire populations but with subpopulations, and we are doing this because of the specific problem we are dealing with, that is antibiotic resistance. As most of you probably know, antibiotic resistance is a problem in public health nowadays, and it will be much worse in the future if we don't tackle it, because many procedures that are now very frequent and normal, like surgeries, will not be possible anymore due to infection. What normally you hear when you hear this thing, when you hear about antibiotic resistance in the news, is that scientists must develop new drugs. But if you think about antibiotic resistance from the population genetics point of view, it is just adaptation. And what a population geneticist can do about antibiotic resistance is understanding how this adaptation is working and trying to slow it down. And that's what we are trying to do. So we are dealing with a specific bacteria that is called Staphylococcus aureus. It's a gram-positive bacteria that is carried by more or less 1/3 of US population. And normally it's in your nose and in your skin, and it's not harmful at all. It just does create some problem when you are in hospital and, for example, you have open wounds, because it's one of the most important sources of hospital-acquired infection. As such, it is used as a proxy to study the infection control protocols in the hospital, because the more Staphylococcus aureus infections you see, the least the infection control is working. And the very big problem is when Staphylococcus aureus become resistant, and especially when it is MRSA, because which means methicillin-resistant Staphylococcus aureus. Why? Because with just this cassette here, the Staphylococcus aureus acquires resistance to an entire class of antibiotics that are the beta-lactams and are the most used in public health. So if you have an infection and you are given the normal beta-lactam antibiotic, the bacteria is adapted to it, so it doesn't die. And then by the time your doctor realizes that the antibiotic is not working and finds a new one, if your health is already compromised, it might be too late for you. The MRSA ecology is quite complicated and not so well known. I mean, we know just one part, that is the part, we know very well one part, that is the part relative to the hospital. Why? Because since it's a big problem, especially when you are in the ICU, what they are doing in England is that when you enter the ICU, they swab you to check if you have the MRSA, and then they swab you every 2 to 3 days during your stay in the ICU, so that they can check if you are infected with an MRSA during your stay in the hospital. And so we know a lot of the transmission and how it works in the hospital, but we don't know much about, or we know much less about how it works in the community. I mean, nobody, you have never seen medical doctor knocking at your doors and asking if they can swab you when you are at home. And also, another thing is that this is all about health. So there is lots of issues about privacy and about not sharing data. So if you want to have to work with data from another hospital or another country, it is much more complicated because of the issues of privacy. And so, although bacteria populations are enormous, and so there is in principle an enormous amount of data, the ones with which you can work is not so big and biased, because you get most from the hospital. Of course, before whole genome sequencing was quite available, still there were methods of genetic typing to identify your bacteria, which were to identify the bacteria that were causing outbreak, and especially to find out whether this, whether it was an outbreak, in the sense that all the people in the hospital were having bacteria that were genetically related. Of course, this is genetic typing is it's based on parts of genes or on the length of microsatellites, so it's much less detailed than using whole genome sequences. For example, in this case, this is an outbreak that was going on in the 2007 in southeast of England. From the genetic type point of view, all the bacteria belong to the same type, that is this MRSA 16 that is up there. But then when we sequenced, we saw that there was much more diversity into this outbreak. And especially if you saw those one in the red, this is the real one that were driving the outbreak, and they were all set in a Brighton hospital. So by using whole genome sequences, you can get much more information. And for example, you can follow the outbreak, because this was following the outbreak, and we were able to study in detail the genotype of the resistance, so if the genes were present and absent. And we were able to find out that due to the public health measures that were put into place to stop the outbreak in 2007, some of the descendants of the outbreak lost their resistance. So using whole genome sequences gives you a lot more information. And so the aim of my work is to provide medical doctors with a tool that, when given the tree of the samples, is able to identify if there is an outbreak, on which clade the outbreak is, and how fast the outbreak is expanding, so that they can understand if this outbreak is serious and how quickly they need to put in place public health measures to stop it. So, for example, if you look at this, yeah, you can see. We can, as this tree, the black one, we can think whether the outbreak is the red one, or whether there are two outbreaks, one into the other, like the red one and the blue one. The red one and the blue one. And yeah, and so we want to have this tool. But we have a problem with language, because outbreak is a very well-defined concept for medical doctors, but it is not necessarily linked to population genetics. There is another term that is more useful from the population genetic point of view, which is clonal expansion. That is a subpopulation that has recently become more abundant and widespread. Yet we are not discussing whether it has swept or not, or whether it is happened due to fitness or whatever. So our model is a coalescent model of a population that at a certain point splits in two subpopulations with no migration, and one subpopulation originates from a single clone here, and for some reason that we are not investigating, it starts to expand. And we want to understand if we can have an effective model of this subpopulation, and if we can use it to infer the evolutionary parameters of the population. So the first attempt that we did was using an exponential expansion. The problem is that here you can see the tree with this is the subpopulation non-expanding, and this is the expanding subpopulation. And you can see that the exponential expansion of the cell lineage does not describe properly the outbreak data. And this is because most probably the Staphylococcus aureus ecology is much more complex than this. One is because there are all these like effects from migration from different from the community and from the hospital, and also because there are probably saturation effects, because you never see a subpopulation that invades. So we tried a more general model that is basically an early expansion followed by a saturation. We used a model that is related to logistic growth. And as you can see here, the tree is much more representative of, much more similar to the real data tree. And that's it, and I will take